mine's too good.
All
I
right.
like this one actually a lot. These are some really good ones. I think my favorite right now is that one.
Which one?
That one right there, the one you just did on.
It's pretty strong. It's pretty indestructible. You can throw that thing down over the stairwell and it wouldn't break. I've got some at home that blow these out of the water.
Oh, really?
Yeah, some of them are super artistic, and I have two at home that are decoration. I've turned them into furniture.
These are really good. Is it all the models from previous year with the juniors or?
No, these are all freshmen.
Oh, they're freshmen. A freshman did better than you.
All right. Good morning, you guys. Good morning, everyone. It's afternoon. It is 12:01. Woo! All right, so I'm going to drop in the chat. The order in which we're going to present. You probably have already seen this. It's the same thing I sent on Remind last night. There you got it. There you got it. We're going to use this order, and we will start about 12:07, 12:08. I'll wait till everybody gets in here, kind of remind everybody of the rules, and then we're going to begin. You guys can ask any questions you want, though, in the meantime. Make sure that you guys can share. Yep, and audio is working. Yep, you should be able to hear. Yep. Beautiful people coming in the room. Good morning, beautiful people. Or afternoon. I don't know why I keep saying morning now that it is the afternoon. Again, there's the link. That's the order that people will be going in. So let's see. I don't even know who's first. Let's see. Looks like Leal, you're going to be first. Don't start yet. Don't start yet. It will be Leal, Abby, Elosa, Brandon, Tubbs, Tracy, Ashley, Alea, Quetta, Sanchez, Hare, Mukhtar, Terry, Quackencherry, Chavez. My plan is to at least get through. Last class, we were able to get through all the way to first period. We got through 17 individuals. So we went through here. See if we can't match that. So we should be able to get through the first 16 to 17 people fairly easily. Need some jams. Y'all need some jams. We're letting some more people in. I'll start giving instructions in just a moment. I'm going to go ahead and drop the link back in here so everybody can see it. Many of you guys got this on Remind last night. Some of you guys did not. That is the link. That is the order in which we will be going. And we'll start in about 1 minute. I'm going to go get my little question bank. I'll be right back. All right, everybody. It is about that time. It is about that time. First person that's going to be going is going to be Dylan. So Dylan, get ready. How this is going to work, again, just to review some of the important things here. There was a couple of people who missed a couple of points. And that was because they weren't looking at the camera. Make sure that when it is your turn, after you've presented your model, that when you're answering the questions, you are looking at the camera. See how my eyes are looking at the camera, but now they're looking at Taylor Tubbs? See how I'm looking at Taylor Tubbs? You can tell the difference. You look at the camera, I assume that you're reading from the questions if you're not looking at the camera, and you just don't score those marks, right? And you want to make sure, 25% of it is just answering those questions. So make sure you do that. When you come on in a moment, you're going to come on, you're going to present your model. Tell us all about your model. Show off. Try not to go over about 2 minutes if you can help it. Try not to go over about 2 minutes when you present, just so you give everybody enough time that we can go. You're the largest class at 32 people in the class. It's quite a few. We want to make sure that we give everybody a fair amount of time, but not too much, so they can show off, but not hog everybody else's time. I will post videos for both classes that you can go back and check them out. So without further ado, your questions are in here. So when it comes time for question, I'm not choosing the questions. I'm going to reach in. I've got 4 beans. And let's say I drew this bean. It will be question number 11. So I'll go over here and I'll be like, do nitrogen bases bond with deoxyribose or a phosphate group? Pretty straightforward question. That's one of the easier ones. You have 3 seconds to answer, and I'm going to have my fingers up. Boom. Once I get my finger down, that's it. You have run out of time. So you've got 3 seconds to answer the question, looking at the camera. Are there any questions before we begin?
Can you repeat the last part, Mr. Pope?
You've got 3 seconds to answer those questions. So I'll have my hands up, right? Or my fingers up. I'll have my hand up and I'll be like 1, 2, 3. I'll answer the question and you've got 3 seconds to answer that question. If you don't answer the question while you're looking at the camera, done. There are 4 questions. 4 questions. And I'll randomly choose them. So last time I drew an 11. This time I drew 13. But we're going to begin. Dylan, share your screen. Take it away. Make sure your camera is on.
Hello. Okay, I'm going to present now. Can you see it?
You can.
Okay. So this is my virtual DNA model. It is this. The flowers represent the phosphate group because of how I just remember like the middle part and how the petals like kind of connect to it. And the duck with the knife is the deoxyribose because when I saw it, because I was searching up things, I just thought of it like as sweet, and even though it's just holding a knife. And then this paintbrush, the brown one, it is cytosine, and the blue one is guanine, and the daisies, the daisies here are just thymine. Oop. And the sunflower is the adenine, and the red ball is supposed to be the 3 hydrogen bonds, and the blue ball is supposed to be the 2 hydrogen bonds. And this is my key.
Excellent. Are you ready for your questions? Oh, can you go back to the model? Let me look at one extra thing. Okay. All right. Make sure you're looking at the camera. Get ready. Here
Okay.
we go. Where is DNA found in eukaryotes?
They are found in the nucleus.
Okay. Does DNA replicate from the 5' to the replication fork? Or if so, explain. If not, explain.
Um. Uh, I don't know that one.
Okay, 25. Why do organisms possess different traits but use the same nucleotides?
They have different traits because of evolution, but they have the same nucleotides because that's how they produce proteins.
Okay, I'm going to extend that a little bit. So they have the same alphabet, but I get different traits, right? The nucleotides are like letters. Why do they have different traits?
Because of evolution and how like they have to survive, so they have to change how they look like and their traits.
What would change the trait? Physically actually change the trait.
Um,
Don't
The-
fret. Let's keep going. Let's keep going. No, it's okay. Let's get to how is chromatin different from chromosomes?
chromosomes differ from chromatins because chromosomes are a pair and chromatins are not a pair.
And there's multiple answers to that. Okay. Thank you very much. The next contestant on the Pope is Right is oh, give that kid a round of applause if you don't mind. What are you gonna do? What are you gonna do? There you go. Oh look, it's Abigail. Is that Abby in the Cheeto bag? All right, Abby, you're next. Let's see.
Can you see my screen?
We can.
Okay. So this is my DNA virtual model project. And first of all, for the bases, I have paper clips because they're numerous in households and they also link together to form chains. So my green paperclip here represents guanine, the purple represents cytosine, the blue represents adenine, and then the pink paperclip represents thymine. I have different color masking tapes. So the green striped masking tape right here represents the 2 bond, and then the blue represents the 3 bond. And I chose masking tape because it can bind things together, but it's still pretty weak. And it can be easily ripped apart with your hands, like helicase rips through the hydrogen bonds.
Excellent.
And then, I then have this Sanrio Purin. It's a dog pudding, and I chose it for my deoxyribose sugar because pudding is composed, like a large part of pudding is sugar, and it's sweet. And then for my phosphate group, I chose binder clips because it's the point of connection between each nucleotide, and it's also more heavy duty, and it can hold more bulk, and it binds things together. And here is my key. Yeah.
Excellent. Here we go. your questions. How many hydrogen bonds between adenine and thymine?
Two. There are two hydrogen bonds.
What are the three components of a nucleotide?
A nitrogenous base, a phosphate group, and a deoxyribose sugar.
What nucleotides are present in DNA?
Adenine, thymine, guanine, and cytosine.
Apparently it likes this question. Why do organisms of different traits, why do different organisms possess different traits but use the same nucleotides for DNA?
Because traits are determined by the order of the nucleotides and not the different types of nucleotides themselves.
Everybody give this child a round of applause, a round of applause. Yes, sir, yes, sir, yes, sir. All right, beautiful. All right, we keep going along. Elosa. What's up, Elosa? I didn't notice Elosa coming in. Hoping that she's here.
I'm here.
Oh, there you are.
It's
Yay.
just being a bit slow.
So Esosa told me that you're able to stay home because your mother trusts you to do what you need to do, and she doesn't trust her to do what she needs to do. Is this true?
Yeah, yeah.
Well, let's just see whether your mother's trust is well placed.
Okay.
Are
All right.
you trying to mock me by wearing gray hair, having gray hair, make fun of me? Is that what you're trying to do? Hurt my feelings?
Definitely.
Oh man. All right.
Okay. So you see my screen?
Yes, ma'am.
Okay. All right. So this is my model. I chose rocks as the phosphate. Oh, this is sort of like to represent a pond, sort of. I chose rocks as my
Ooh, you gotta change your slide.
phosphate.
We're on slide one, the title.
Change it? Oh, on my, oh, okay. You see it now?
See. Oh yeah, perfect.
Okay. Okay. So I chose rocks as my phosphate groups because in like ponds that I've seen, like in the DNA, the phosphate groups are around the like the nucleotides and stuff like that. So I chose rocks, and then I chose lily pads to be my hydrogen bonds because although lily pads do hold sometimes like frogs and different like organisms like insects, it's not really like durable. And I chose different types of frogs that are showing, they're just like angry frog, smiley frog, to show like the different nucleotides. And
what about for the deoxyribose sugar? What is that?
those are different types of koi fish.
Oh, there's three fish swimming together.
Yeah, the amount doesn't really mean anything. It was just it's in part of some ponds, so I decided to put it in there. Yeah.
Okay, let's check out that key.
Okay. So this is my key. Like I said, the rock is the phosphate group, and the koi fish are the deoxyribose. The angry frog is thymine, and the hungry frog is cytosine. The cowboy frog is guanine, and the yellowish lily pad is with the flower on it is 2 hydrogen bonds, and the one with the pinkish flower on it is 3 hydrogen bonds, and then the smiley frog is adenine.
Can you go back to the model real quick? That tells me which one which bond should be with which nucleotide or nitrogenous base.
Okay.
Coolio. All right. Are you ready for your questions?
I believe so.
All right. Yeah, I guess it doesn't matter. It's happening one way or the other, right?
Yeah.
Okay. So, wow. Where is DNA found in eukaryotes?
It is found in the nucleus.
Where is DNA found in prokaryotes?
It is found in the nucleoid in the cytoplasm.
Which structures are present in, no, excuse me. Sorry. Which nucleotides are present in DNA?
The nucleotides that are present in DNA is adenine, thymine, cytosine, and guanine.
How is chromatin different from chromosomes?
I believe chromatids make up chromosomes, and yeah, chromatids make up chromosomes. And chromosomes are just composed of chromatins. Like chromatins are like a smaller versions of chromosomes.
All right, everybody give this child a round of applause, Beautiful, beautiful, wonderful, wonderful. Ooh, it's Friday, hey. All right, let's see. Next contestant on the Pope is Right is Brandon Osamba.
I'm next, sir? Okay.
You are next.
Okay. Can you see my screen?
Hang on.
Can you see my camera?
I cannot. That's a good question. Let me see if I can find you.
Can
Oh,
you see
there
my-
you are. Woo! Okay, now you gotta share your screen. We're good.
It says disabled. Okay, now I got it. That's fine.
Say again.
No, I think I got it.
Okay, there you go.
Can you see my screen?
We can.
One sec.
Yeah, you were good.
Can you see my screen?
Yes.
Okay, cool. So this is my biology project. This is my map. For the phosphate group, I picked the soccer ball as it holds the system together, similar to how a soccer ball holds up soccer. Without a soccer ball, there is no soccer, essentially, similar to phosphate group. For my deoxyribose, I chose Gatorade because it's similar, because just like deoxyribose, it's sugary and it's a sugary food. It gives players energy. You drink a Gatorade, you feel more energetic. It has sugar. For my guanine, I picked the Man City logo, which is a team. And guanine is a nitrogen base, and I decided to pick it. And I decided to pick the Man City logo for guanine. For cytosine, I picked Man United because it pairs with Man City, as in the real world, Man City and Man United are city rivals. And yeah, for my thymine, I picked the Liverpool logo because it pairs with adenine. And for my adenine, I picked Everton as it pairs with my Liverpool logo, as in real life, these tend to be city rivals. And last but not least, my whistle, or my 3 hydrogen bonds, I picked the whistle. I picked the Premier League trophy.
Okay, so which one's which? Which one's two
And
hydrogen
for
bonds?
my 3 hydrogen bonds are the trophy, and then my 2 hydrogen bonds are the whistle.
Roger
And I
that.
did the whistle for the hydrogen bonds because it keeps the game in control, similar to the 2 hydrogen bonds. And for the 3 hydrogen bonds, I put the Premier League trophy, as it's very rare, important, and wanted.
Okay, let me ask you a question. Liverpool and Everton, which is the better team?
Oh, I mean, that's a super obvious question. Everton.
You know, my ancestors immigrated from Liverpool.
Really?
Yeah, and Liverpool's the best. What are you gonna do? What are you gonna do? I'm just kidding. All right, let's see the key. That is true. Though.
soccer ball, phosphate group, Gatorade, deoxyribose, guanine, Man City logo, cytosine, Man United, thymine, Liverpool, adenine, Everton, the Premier League trophy, 3 hydrogen bonds, and 2 hydrogen bonds is the referee whistle.
Okay, get ready. You ready for your questions? And man, does it like
Sweet.
this particular question. Good grief.
Okay.
Where is DNA found in eukaryotes?
I believe it's found inside the nucleus.
Does DNA
eukaryotic
transcribe?
cells.
Does DNA transcribe from the 3' to the replication fork? If so, explain. If not, explain.
DNA polymers are only able to build new strands from the 5 to 3 direction, I believe, which is due to the fact that DNA polymers only add DNA nucleotides to the 5 to 3 direction, 3 to 5 direction.
Okay, you got to be looking at your camera. The camera, there you go. Okay, 14. What is the monomer for a polymer of DNA?
A nucleotide, I believe.
What percentage of organisms on Earth use DNA as their genetic information?
I think all animals use it because we all share a common ancestry.
Okay, what about organisms, period? Not
Yes.
just animals.
Yes. We all share a common ancestry.
Okay. Thank you very much. Thank you very much. Everybody give this child a round of applause, We love this child. What are you gonna do with this guy? What are you gonna do with this guy? All right, the next contestant on the Pope is right. Hold on. Oh, yeah, they can hear you, Cotton. Let's see, Taylor Tubbs. Woo, Taylor Tubbs. What happened to my screen?
Can you see it?
Not because something you did, but because something I did. Okay, now I can see it. Yeah. Okay, Tubbs, take us away.
This is my virtual model. So the Tic Tac represents the phosphate group, and the egg represents the I don't know how to say that, D-ribose. Deoxyribose. Well, I realized I should have put the egg as the phosphate and the Tic Tac as the D-ribose, because Tic Tac is like candy, sugary, you know. And for the 4 bases, I just use the brands for the chapstick brands. So this one represents adding, and this one represents thymine. And the light one represents cytosine, where the darker one represents guanine. The dime represents the 3 hydrogen bonds, and the penny represents the 2 hydrogen bonds.
Okay, and where the penny is, go back real quick. Where the penny is, are those nitrogenous bases? Is that guanine and cytosine or adenine and thymine?
This is cytosine and guanine.
Okay, got it. All right, here's your questions. Let me see, I got it. How many base pairs does it take to transcribe 1 codon?
3.
Boom. What are the 3 components of a nucleotide?
Nitrogen base, phosphate, and sugar.
What sugar? There's a lot of them.
I don't know.
Okay, don't worry. Don't let it throw you off. Why do organisms possess different traits but use the same nucleotides for DNA?
To determine the traits they get from their parents.
Right. So why is it we all inherit the same nucleotides, but we have different traits? What's different?
I don't know.
Okay. Is DNA replication conservative, semi-conservative, or dispersive?
Semi-conservative.
All right, everybody give this child a round of applause, a round of applause. It's the great Taylor Tubbs, yeah. You got a fan in Medina. You got a friend in me. You've got a friend in me. Oh, Taylor Tubbs got a friend in Medina. All right, let's see. Tracy Tarleton. Tracy Tarleton.
Hi.
Hi.
Okay, I'm gonna share my screen.
Medina says you don't have a friend in him. Only Taylor Tubbs does. He doesn't have enough room.
Yeah.
He doesn't have enough room in his heart for two people.
Oh. Okay, you can see my screen, right?
Yes, we can. And I can see you for one of the few times I've actually seen you. I know your
I know
voice.
you haven't seen me since the beginning of the school year.
I know it. It helps to actually lay eyes on people. Jesus. Okay.
All
Let's go.
right, so this is my virtual DNA model. So what I did, because it looks like a face, but anyway, so what I did is I used human body parts, which is ironic because we're talking about DNA. Yeah. So the yellow bone and the yellow bone, it's cytosine. Guanine is the white one. Red is adenine. Blue is thymine. Deoxyribose is the mouth. Phosphate group is the eyes. The brain is 3 hydrogen bonds, and the heart is 2. And I made an explanation slide. Oh my gosh.
You got a 5 and a 3 in, okay. Holy smokes. Oh yeah, I'm gonna see your explanation. Let's check out your explanation slide.
Okay. So the bases of the bones, and the reason this is because bones make up our body and they make us stand. It has a structure, and without the nucleotides, then there would be no structure for the strand of DNA and it wouldn't hold it up. Deoxyribose is the mouth because the mouth includes the tongue, and the tongue has taste buds, and that allows you to taste sugar. Phosphate groups are the eyes because there's 3 main parts of the phosphate group, and there's also 3 oxygens around the phosphorus. And there's 3 main parts of the eyes. The hydrogen bonds are the brain and the heart, and I couldn't decide which was the most important organ, but I decided on the brain because it holds the most information, and that's why it's 3, because 3 is more than 2.
Gotcha. Yeah, I don't know how you can't live without a brain nor a heart. So what are you gonna do? Look at those skeletons. What is that? Was that a Popeye cartoon? Woo! Okay, get ready for your questions. Oh, I didn't draw them yet. How many hydrogen bonds between adenine and thymine?
2.
Which nucleotide is absent in DNA?
Uracil.
Where the heck? Oh, here it is. Are mutations always deleterious? If not, how else could they be categorized?
No, they are not, and they can be categorized by positive, neutral, damaging, and lethal.
Ooh, you're the only kid all day that's got that one. Jesus, okay. How is a prokaryotic chromosome different from a eukaryotic chromosome?
Prokaryotic chromosomes are found in the nucleoid, which is in the cytoplasm, and eukaryotic are found in the nucleus.
Okay, and I'm gonna go ahead and not freak everybody out. There are multiple answers to that question. So if you've memorized a different answer, that's okay. As you've seen on the rubrics on the IB exams, there's multiple ways to be right. So that was one difference, and that was it. So thank you very much. Everybody give Tracy Tarleton a wonderful round of applause. Yay. Medina said he made room in his heart for you, but he has no room for others. He can handle 2
Thank
people
you,
at a time.
Matthew.
Matthew Medina, what? Oh my God. All right, so let's see who's next on The Pope is Right. There's room for who?
Dylan.
Okay, there's room in there for you too, Dylan. Ashley Matlock. Ashley,
Hi.
hello. Oh, look at that shoe organizer.
Don't pay attention. Okay.
Ooh, you got animals. I love animals.
Yep. Okay, so for my DNA model, I decided to use animals except for the deoxyribose. And for the phosphate group, I used tree trunks. And then for the deoxyribose, I decided to use Skittles, red Skittles. And for guanine, I decided to use well, for the nitrogenous bases, I decided to use different colored wolves. So for guanine, it's a gray wolf. Cytosine is a black wolf. Adenine is a brown wolf, and thymine is a white wolf. And for the 3 hydrogen bonds, I decided to use a deer. And for the 2 hydrogen bonds, I decided to use a rabbit. And this is my keys.
Okie dokie. Are you ready for your questions?
Kind of scared, but
Don't be scared. Hey, being scared is a good thing because the more often you're scared, the quicker you get over it. The quicker you get over it, the easier it is to move through life. You can never get over fear if you've never been afraid. So let's see. Okay, 14. What is the monomer for a polymer of DNA?
I say
Oop, gotta
nuclear.
look at the camera. Gotta keep your eyes on camera.
Oh, sorry. I said nucleotides.
Okay, 15. Which nucleotides are present in DNA?
Guanine, adenine, cytosine, and thymine.
I like this question, to repeat. Are mutations always deleterious? If not, how else could they be categorized?
I'm not exactly sure about that question.
Okay, 26, keep it cool, keep it cool. How is chromatin different than chromosomes?
I said chromatin is a lower order lower order of DNA organization where chromosomes are higher order of DNA. Organization, sorry.
Okay, technically, you get credit for that. Can you tell me how it's more organized? I'm curious. It's a follow-up question. No stress.
Let's see.
If you can't, that's okay. But you ought to follow up on it because if you were technically, you got that point for sure. On the exam, you might have to talk about that a little bit about how that organization is occurring. All right. In fact, I'm 100% certain of that. That's why I asked that follow-up question. Hint, nod, nod. Hopefully, all you guys are hearing this. Let's give this kid a round of applause. Yes. Ashley Metlock, she's a wonderful child. In a room with no lights on, saving us her carbon footprint. She's decreasing that carbon footprint for us. I appreciate that. Natural light. Her room looked comfortable. Did you notice that? Kind of soft lighting. If I went in there, I might just fall asleep. Like, oh, hold on, Ashley, I gotta take my shoes off, put them in there, and go to bed. I'm tired. Looks like you had something to make a mosquito net out of too. Okay, so next contestant on the Pope is Right is going to be the wonderful, the lovely Earl. No, I take that back. It is Donalea Lopez. Yes.
Okay, I'm gonna go ahead and share my screen.
Yeah, Lopez. Earl's cheering you on.
Hold on, I'm trying to figure out. Okay. Can you guys see me and my?
We can see you.
Okay. Oh, I'm gonna go ahead and click.
I'm gonna get in that little hammock in the corner and go to sleep up there like one of those little elves that they have that watches their house in Christmas time. Okay.
This is my DNA model. Can you guys see it?
We can see the key.
Oh my gosh. Did it still show the key?
There it is. Look at that.
Oh, hold on. I'm sorry.
Ooh.
I'm sorry, it's so slow. I'm on the Chromebook.
Don't worry about it.
Okay, there. Okay, so this is my DNA model. For my theme, I decided to go with fruits and vegetables. For my phosphate, it's cheese. My deoxyribose, it's mango. My cytosine is asparagus. My guanine is a carrot. This is my thymine, it's a pickle. And my adenine is a celery stick. The tomatoes, it represents my 3 hydrogen bonds, and the oranges are my 2. For my hydrogen bonds and for my nucleotides, I just chose these out of spite. I didn't really have a connection for these, but for my deoxyribose, I chose a mango because it's high in sugar, and a deoxyribose is a sugar. And for my phosphate, I chose cheese because cheese or dairy in general is a food high in phosphorus.
Okay, yeah, there was a one caveat to this. You're like, I got fruits and vegetables, but I got to get my little shot of dairy in there. Yeah. Okay, good deal. Are you ready for the questions? It doesn't really matter if you're not, because you're going to get asked them anyway. Where is DNA found in eukaryotes?
It's found in the nucleus.
Do nitrogen bases bond with deoxyribose or phosphate groups?
They bond with deoxyribose.
Which nucleotide is absent in DNA?
Uracil.
Why do organisms possess different traits but use the same nucleotides?
For DNA, it's due to the order of the nucleotides.
Okey-dokey, Finoki. Everybody tell this soccer player that she did a great job. Great job. Hey, is Everton or Liverpool better? Who's better?
Of course.
Liverpool, of course. Liverpool. That's right. That's right. Little trivia. That's where my ancestor. Just to take a second, I have a docket on a ship that whoever my ancestor was, we don't know their first name. They were illiterate. They signed a docket on the ship to come to the United States. They left Liverpool. We do not know their first name because they were illiterate, and they signed their name with an X, and they wrote and then whoever was I guess the sailor that was logging them on the ship wrote out the last name next to the X. But I know that that's where they boarded the ship. Okay. Let's see. That's on my mother's side anyway. Argueta, Jessica, tell us all about it. Oh, there she is. She was ready. She had her camera on. She was like, I'm ready for this. I got this. I own this.
Okay. Can you see my screen?
We can.
Okay. This is my virtual DNA model. So I use like random objects. I didn't have like a theme like other people. I use dice. It shows 3 and 2 as the chromosomes. And then I use Legos because they connect just like the bases. The green is cytosine, the pink is guanine, blue is thymine, and red is adenine. I used the M&M because it's a sugar, just like the deoxyribose, or however you say it.
Deoxyribose.
And then I use bottle caps just because I guess they're solid, like phosphate.
Phosphorus has no gas.
This is my key.
You got a Red Hot Chili Pepper sign over here on the right side.
What?
That's the Red Hot Chili Peppers symbol, isn't it?
Oh, I don't know.
Romello might know. I know he kind of I think he likes chili peppers.
Yeah.
Is that the symbol?
Yeah, it is on one of their albums. Yeah.
Okay. All right. Are you ready for your questions? Let's see. 1, 2, 3. Okay. Oh, these are new. I haven't seen some of these. She's like, oh God. Okay. In the backbone of DNA, what joins deoxyribose to another molecule of deoxyribose?
The phosphate group?
How many nitrogen bonds between adenine and thymine?
It's 2.
Are mutations always deleterious? If not, how else could they be categorized? Look at the camera. Look at the
oh.
camera.
No, they're not always. It can be insertion, I think it's called.
All right, and is DNA replication conservative, dispersive, or semi-conservative?
Semi-conservative.
Give this child a round of applause. Yes, yes, yes. She's conserving electricity just like Ashley Metlock. Excellent, excellent, excellent. All right, so let's see. The next contestant on the Pope is Right is Earl. Earl gets 50 points off just because his name is Earl. I love the mess of Earl. There's Earl. He's in the boardroom. And he's chewing that gum that he got from before. Okay. All right, Earl, I'll stop picking on you. Go for it.
I need Jessica to stop screenshot.
There you go. He's in the boardroom.
Okay, can you see my screen?
Yep, hold on a second. I dropped your question on the floor. Okay, yep, we can see it.
Okay. Hello everybody, my name's Earl and this is my DNA model project. And the theme that I chose to go with for my model is sports themed. As you can see, here's Dwayne Johnson, The Rock, as the phosphate group. This is a baseball base. If you guys know baseball, after you swing the bat and hit the ball, you're supposed to run to it. And I chose this because
I thought you were supposed to run away from it and then run back to it.
I think so. You run and then you touch it.
Yeah, after you, I'll pick on you later. Go ahead, baby.
Okay, so the balls, the different types of sports balls, those are the cytosine, guanine, adenine, and thymine. And yeah, you can see everything is connected and linked up with the double hydrogen bonds and the triple hydrogen bonds. And yeah, here's my key if you needed, this thing's in the way, but if you needed to, if you couldn't understand what I'm saying, I'm sorry.
No, I could understand everything.
So my explanation for the things I've chosen, like I said, Dwayne "The Rock" Johnson, he was a former WWF/WWE wrestler. So, and his nickname, The Rock, relates to like phosphorus because phos, like the prefix phos, and or I don't know. And then the baseball base, I already explained that because they share the pentagonal-like shape. And the reason why I chose the golf and the soccer ball for cytosine and guanine is because when you in the sports, soccer and golf, you're not really supposed to touch the ball with your hands. You use your feet in the golf club. So that's how they're related to each other. And then basketball and volleyball, I chose them as adenine and thymine because you can touch the ball. And the 2-pack of Gatorade bottles I chose to represent the double hydrogen bond for adenine and thymine is because when you think of hydrogen, you think of water, H2O. So I thought that'd be cool to do it. And athletes, they need water and Gatorade to stay healthy. And the same thing for the hydrogen bonds in between cytosine and guanine. And I just added an extra Gatorade thing. And yeah, that's my presentation. Thank you for watching.
Okay, are you ready? How many, speaking of hydrogen bonds, how many hydrogen bonds between guanine and cytosine?
There are 3 hydrogen bonds between guanine and cytosine.
Beautiful. How many base pairs does it take to transcribe one codon?
It takes 3 base pairs.
Okay, they gotta look at the camera, yo. 14. What is a monomer of a polymer of DNA?
Nucleotides.
Which nucleotide is absent in DNA?
Uracil.
There you go. Everybody loves or Eral. Eral, everybody love him. Woo! Y'all give him some claps. Claps. That guy needs some claps. See that guy? So he won. Well, how much money was that you won in advisory? Was it $10? And he's still chewing that gum. Jeez.
Yes, sir. I'm not gonna let that gum go to waste.
Don't let it go to waste. That would be wasteful. Ashley Matlock and Taylor Tubbs have already showed. No, who was it? Argueta, and I think it was Tubbs too. They're all conserving electricity by not having the lights on, so therefore there are less CO2 is going in the atmosphere. So let's keep up that theme. Let's keep up that theme. Not be wasteful. Let's see. Next is going to be Hare. It's Tabitha Hare. Woo!
To be me indeed. All right.
I'm
Can
actually
you
coming to
see
your
my screen?
neighborhood in about 3 hours, 3 or 4 hours. Get ready.
Wait, why you have to go in my near? Okay, that's concerning.
Yeah,
Can you see
well,
my?
it's not because of you. All right.
Okay. But can you see my screen?
Oh yeah, absolutely.
Ready. So I know you've already seen mine, Mr. Pope, but I'm still going to present it.
Right.
So, for my DNA model, I didn't have a general theme. I just kind of did whatever made sense to me. So for my phosphate group, I did bricks because a lot of bricks or rocks are made out of phosphate. And then for deoxyribose sugar, I did sugar cubes because, well, it's a definite shape instead of just a random mound of sugar. And then for the bases, I did different brands of soda. So for amine, I did Dr. Pepper, and then for thymine, I did Mr. Pibb because Mr. Pibb is often seen as the off-brand version of Dr. Pepper. And then for cytosine, I did Sierra Mist, and then for guanine, I did Sprite, which again, Sierra Mist is basically the off-brand version of Sprite. And then for my hydrogen bonds, I did silly string because silly string is weak. And so for the, hold up, I messed that up. But anyways, for the Mr. Pibb and the Dr. Pepper, I gave specific amount for how many bonds it's supposed to have. So I accidentally switched it, but that's okay. So this one's supposed to have.
No, go ahead. I'm sorry.
Okay, and then this is my key. For covalent bonds, I just had a black line, and then for hydrogen silly string.
The thing that was funny for me was that whenever I saw this before, I was looking at it through my telephone, and I thought that the silly string was like a glue stick, and it would work there as well too. So that was like my comment to you. I was like, "Oh, I love the glue sticks" or whatever, and you were probably thinking, "What's this guy smoking? What's wrong with this guy?" Okay, good deal. Are you ready for your question?
No, but I don't have a
No,
choice.
what are you
So let's
gonna
go ahead
say?
and go.
Yeah. All
Let's
right.
get this over.
So, do nitrogen bases bond to deoxyribose or a phosphate group?
Deoxyribose.
All right. Why must DNA synthesis result in identical copies of DNA?
It has to, because if the daughter cells have different something, then it could occur in mutations.
What occurs to phenotypes when two dominant alleles are paired?
It's called codominance, and both traits are expressed.
24. What is the term for an organism possessing the same two alleles for a gene?
Homozygous. Did I pronounce that right?
Yep, homozygous. They are homozygous. Okay, well, I really wasn't trying to confirm right or wrong, just because, you know, give it away for the next person. But thank you very much. Thank you very much. Everybody give her a round of applause. The wonderful, beautiful hair. Yeah. No, I think I'm just coming. There's a takeout by your house, and my wife told me I need to go over and pick it up after work. She wasn't cooking even today, so. Okay, that's why I'm headed that way. Let me see. The wonderful, the marvelous Mukhtar. Hello, Mukhtar. Oh, Erel got kicked out. He's trying to come back.
Can you see my screen?
I'm sorry?
Could you see my screen?
Oh, yeah, we can see it.
Okay, so this is my DNA model.
Mhm.
Okay, so for the ice cream scoops, I used it's the phosphate group, and the cones are the deoxyribose. Strawberry ice cream is cytosine based. Chocolate ice cream is adenine based. Vanilla ice cream is guanine, and then sprinkles are the 3 hydrogen bonds. The chocolate chips are the 2 hydrogen bonds. I just put ice cream theme. That was just it. And then this is my key.
Okay, follow-up question. Of those 4, is guanine, cytosine, thymine, or adenine, which ones taste the best?
Guanine.
Really? You're a vanilla fan?
I don't like chocolate.
You don't like chocolate? How do you not like chocolate? You fail. Where do you work again? Do you work at an ice cream place?
No, I work at Salata.
Oh, that's right. That's right. Salata. Yeah, well, they don't have this at Salata, man. They'd throw you out if you came in there and started eating ice cream like that. They're like, what? That runs against this. Oh, I likes that place too. Okay, so let's see. All right. Questions? Let's see. I have witnesses here to testify that these questions are random, in case any of you guys are wondering. Okay, which nucleotides are present in DNA? You'll never guess this one.
Wait, what was the question?
Which nucleotides are present in DNA?
Adenine, guanine, cytosine, thymine, and yeah, that's it.
Okay, yeah. All right. Why must DNA synthesis result in identical copies of DNA?
Oh, this is the one I didn't know, but
Time. Okay, don't let it throw you off. Don't let it throw you off. Take a deep breath. Okay. Why do organisms possess different traits but use the same nucleotides for DNA? Just take a deep breath. One more question. What is the enzyme responsible for DNA synthesis? So we're talking about putting the nitrogen bases together.
you said what is the enzyme?
Which enzyme puts the nitrogenous bases together? Don't freak. It's okay. It's all right. Still, you did great. Everybody give this child a round of applause. Give her love. Send her messages of love and rain flowers down. And send her vanilla ice cream because she doesn't like chocolate. Sounds like somebody else I know in my life, which is great because that just leaves more chocolate for me. That leaves more chocolate for me, yo. All right, so let's see. Man, how can you, what are you talking about? Chocolate ice cream is delicious. It's fantastic. Okay, the next contestant on The Pope is Right is Romello Salinas. Romello Salinas, everybody give Romello Salinas a warm welcome. Romello looks like he's flying through space on some sort of spacecraft. If you see him on video, it's like a party back there, doesn't it? Okay. Okie dokie. Let's hear from Romello. We can't hear you, baby.
Oh, sorry, I was muted. Okay. So I decided to make a 3D model. Try to at least try to do it, but I couldn't like use other materials, so I had to use shapes. Like, so for the key for my project, orange cylinders were guanine, green cylinders were cytosine, yellow cylinders were adenine, and blue cylinders were thymine. And then the spindles are very small and they're in the middle. I colored them. They're the hydrogen bonds, and the red spindle represents 2 hydrogen bonds, and the black represents 3 hydrogen bonds. And then on the outside, the small pink spears represent the phosphate, and the big white spears represent the deoxyribose.
Okay, get ready for your questions. Get ready for your questions. All right, so we'll do this in order. Where is DNA found in prokaryotes?
Nucleoid.
Whew, freaks me out. How many nitrogenous bases are between each groove in the double helical structure of DNA?
10 to 10.5.
Ugh. Where is that? Oh, here it is. Are mutations always deleterious? If not, how else could they be categorized?
No, they're not always, and they can be classified as positive or negative.
Joke. And finally, how is chromatin different than chromosomes?
They're made with a special protein. I don't remember the protein's name, but I know they're made of special proteins, and I could be smart and just say the name.
Which one is it? Chromosomes or chromatin has the protein?
Chromatin. It's made with a special protein.
Okay, thank you very much. Thank you very much. We glad you played. Everybody, this round of applause. All right, let's see. Stein, I've got a stein in my eye. Where's Stein? Yeah, you know, you get a stein in your eye, it hurts like crazy. It's a stein is it's an infection that's subdermal, that's under your skin, and they happen in the corners of your eyes, and it hurts like crazy. Okay, I don't see her, so let's go to Aspen Jenkins. Aspen Jenkins, I want to hear from Aspen Jenkins. Come on, Aspen Jenkins, come on down. You're the next contestant.
Can you hear me?
Am I missing somebody? I think I skipped several people. I can't see. Yeah, Aspen, I'm sorry, I skipped other people, but go ahead since you're there.
Wait, can you hear me?
Yeah, I can hear you fine.
Is it okay if I can go next class? Because I'm not done with mine.
Yeah, well, technically, I did skip some people. I'm still recovering from that surgery. And I'm looking, I should put this on my right side. So who did I miss? Oh, I should have gone to Terry after Mukhtar. Terry, quite contrary, how does your garden grow? Tell us about it. All right, I'm going to look through the computer, and she's got a model, a physical model, so we'll all look at Terry. Oh yeah, Terry. So what you can do to see her, I don't know if I pin my screen, if it will work, but if you go to the upper right corner of your screen, you can pin her image. Woo! There she is. Look at that. All right. What's up, Animal Cell?
Okay, well, I built my model. So I have the key right here. So for the yellow popsicle sticks, they're adenine. For the red ones, they're cytosine. For the blue ones, they're guanine. And for the orange ones, they're thymine. For the green pipe cleaner that's twisted with the red one, those are my phosphate group. And then the red pipe cleaner is my deoxyribose sugar. And then I don't know if you guys can see, but on the hot glue, I did the hydrogen bonds, and I wrote 2 for the adenine and thymine and 3 for cytosine and guanine. And yeah, that's it.
Okie dokie. Are you ready for your questions?
I guess.
What you guess? What you gotta do? You gotta be ready. We're ready. All right, Terry, quite contrary. Which nucleotide is absent in DNA?
uracil.
Yes. Why must DNA synthesis result in identical copies of DNA?
So they don't have mutations.
Okay. How is a prokaryotic chromosome different than a eukaryotic chromosome?
And eukaryotic chromosomes are found in the nucleus, and the prokaryote is found in the site, the nucleotide.
Okay. And 26, how is chromatin different than chromosomes?
Chromatin is like DNA and proteins that make up chromosomes.
In chromosome?
Huh?
Maybe I only heard half of it.
Oh, chromatins are the DNA and proteins that like make up chromosomes.
Okay. Everybody give that child a round of applause, and I'll pick that up after class, or you can leave it on the table there, the lab station. Let's see, the next contestant on the Pope is right. Let's see who that is going to be. Oh, come on, silly thing. We might only have time for one more, potentially two. I went Tabitha. Chavez, it's Chavez, the wonderful Chavez. Let's hear about it, Chavez. Yes, sir.
All right, here we go. So let me share my screen. Photos, share. Okay, so I decided to use my art styles of working the actual DNA of final. I call it the DNA file mix. I got the title from Kingdom Hearts file mix. So let me see if I can zoom in so you can see better. Okay, so I organized as a keyblade. Cytosine is the original keyblade. Grandmine is the Dust Keeper of Keyblade. Annie is the Bond of Flames, and Thymine is the Rumbly Rose. The base pair is actually the Heartless, because the reason why I chose a Heartless because I was playing this Kingdom Hearts and there was this Heartless swarming around, swarming, and I was like, oh, it's kind of looking like a DNA model, and that got me an idea. So that's where I kind of drew this. The heart is represented by bonds, and the nobody symbols is 3 bonds. Huh? 5. That's what I said. I forgot to put a hydrogen bond, so I just put 5 hydrogen bonds. And nobody just 3 hydrogen bonds. All right, let's do it.
In the backbone of DNA, what joins deoxyribose to another molecule of deoxyribose?
The sugar molecule.
What is the name of that molecule?
Deoxyribose, is it? I think it's what the name. I know it was a sugar molecule, but that's what I know.
What are the rules for base pairing?
The 1:1 Stromatoni ratio. That's what I remember the rules of base pair.
How many base pairs does it take to transcribe one codon?
Oh, is it 3? Yeah, 3. Oh, say again.
You asked a question, or is that an answer? 3?
3.
Let's see, 19. What is up with this? Are mutations always deleterious? If not, how else could they be categorized?
Okay, that was a hard one, but I know not all mutation or deletation, because there's other effects like harming, but there's like substitution, telomeres, and no mutation. That's what I can answer.
Mr. Pope, we can't hear you. We still can't hear you.
Hold on. Hello? Hello? You can hear me now?
I can hear you now.
Woo! Okie dokie. Okay, it is going to be Emma Masisi. Emma. Emma, hello. Oh, I saw a letter in class. I thought I saw her today.
She's in.
Oh, you see her?
Yeah.
Hello. Oh, there she is. You can share your screen. All right, there we go. Let's see. Hey, I think what you did is you need to share the application, not the desktop. I think what you did is you shared your desktop.
I don't know if it's working.
Share the application because we could see it before. It's like you were sharing your screen and then you shared your screen on top of sharing your screen. There you go. Let's start over. It'll be like share application and then share screen. Emma, I am going to run out of time. So we're going to pick up with you. We're going to pick up with you when we come back. I'm going to let you guys go. The bell's going to ring in about 30 seconds. We will pick up and finish with everybody on Tuesday. Hope you guys have a wonderful, wonderful weekend. Play in the street and have a good time.
Enjoy your weekend.
And I'll post this on the website soon so that you guys can see it. Play a little exit music.
Good job, everyone.
Good job, everybody.